So we're going to continue with carbohydrates, and so we've talked about monosaccharides and disaccharides, and I was actually reading an American Chemical Society, I don't know if it counts as a blog, but on LinkedIn this morning about whether high fructose corn syrup is bad for you. And so I learned a lot about high fructose corn syrup. Okay, so have you guys had that discussion elsewhere? You've heard in the news, like, you know, is it bad for you? Is high fructose corn syrup and everything making America obese? Have you guys heard that debate? And so what is sucrose? Here's another little quiz. You don't have to draw the structure, but what is sucrose? A monomer, a monosaccharide, or a disaccharide? Disaccharide. A disaccharide of what monomer building blocks? Glucose and fructose. Okay, so you have equal amounts of glucose and fructose because it's a 1-to-1 ratio, right? And I confess I didn't know this until I read this blog this morning. Does anyone know what high fructose corn syrup is? Claire, what is it?
Processed corn mixed with like sulfuric acid.
Yeah, so I don't know if it's mixed. Maybe there's some acid in the processing, but it's basically corn syrup, corn sugar. So the corn sugar.
Yeah, I think that's how you make the sugar.
Well, so it starts as glucose. Okay, so so they extract glucose from corn, and then enzymatically and chemically convert some of it to fructose. Okay, so now we have a combination of glucose and fructose. What does that sound like? Sucrose. Okay, so table sugar, if we put, I can't stand putting artificial sweeteners in my coffee. I have to use real sugar. So if we put table sugar in something, it's sucrose, equal amounts glucose and fructose. High fructose corn syrup is where they've converted some of the glucose in corn syrup into fructose. And so if it's equal amounts, you might say, what's the big deal? You know, so our enzymes are going to break down sucrose into glucose and fructose. So if we just have high fructose corn syrup, if it's half and half, what's the big deal? So it turns out that it's not half and half. So in the conversion, it's more glucose than fructose, and as we'll learn about in metabolism, glucose is what triggers insulin and, you know, tells your body, oh, I got plenty of sugar. So if you have more glucose in your diet, your liver can turn it to fat very, very easily. Okay, and so
That's true.
it may well be true. Yes, in fact, you know, there's certainly people on one end of the spectrum that say only eat things that come from a plant that you recognize. Okay, so if you're gonna eat sugar, eat an apple, you know, eat an orange. You know, I still like my cake and Krispy Kreme, so we're not gonna throw that out. But if you have a lot of soda and things that's made sweet by high fructose corn syrup, it's not the same as eating an equal amount of sucrose, because the ratio is not what it is in sucrose. Plus we have enzyme, you know, plus things like fruit have fiber in them to make us feel fuller. You know, so if you're consuming a lot of calories from high fructose corn syrup and juices and processed foods, you get a lot of calories without being full, and that's a bad thing too. So I learned something. Maybe you did too.
You said there is less than half fructose?
Right. So even though the name says high fructose, it's kind of a trick in that there's a lot of glucose and probably even more than half glucose. So it's trying to mimic sucrose, but not quite getting it right, is what is the science I've read.
What's sucrose?
Sucrose is half and half because it's a disaccharide of one of each. Yep, and we have enzymes that break it down, and so then we get one of each, but usually it comes with things like fiber and to make us feel full. Okay, so that's monosaccharides, disaccharides. So we're going to talk about polysaccharides today. And so another word for polysaccharide that maybe you may or may not have heard of is glycan. Okay, so just another word. And so these polysaccharides are used for stored fuels. Okay, so we're going to talk about starch and glycogen today. And they're also used for structure. Okay, so they're both energy and structure. And so we can look at these polysaccharides as homopolymers, so all one monomer unit, or heteropolymers, so more than one unit. And this makes it look like they're always regular repeats, but that's not always true either. And then either homopolymers or heteropolymers can be linear or branched. Okay, so we'll talk about different examples of that as we go along. So the stored for fuel, so the stored polysaccharides can be either starch or glycogen. And where would you find each of those? Where is starch found? In plants. So it's the plant form of stored sugar. Okay, and where is glycogen found? In animals. So it's the animal form of stored polysaccharides. And in fact, both of these are homopolymers of glucose. So it's just glucose monomers linked together, one way or the other. We'll talk about how they're different. So it's just glucose in a polymeric form, and the starch is the plant form of that, and glycogen is the animal form of that. And so where in animals is glycogen found? Where in us? Liver and muscles. Most of it is in the liver. A little bit of it is in the muscles. Okay, and so one of our favorite cartoons at our house is Baby Blues. Okay, in case you can't read that, so the dad's saying, "Eat your green beans." I'm not hungry. Not hungry. Those things make you strong. Really? So let me explain how. So he's going to do a little bit of daddy metabolism. As you chew, your saliva is mixed with the beans and lubricates the food as it begins to break down the starches. So there's the starch connection, right? When the chewed up beans or bolus hits your stomach, it's liquefied and squirted little by little into the small intestine. There are enzymes and secretions from the pancreas and gallbladder. So what's in the pancreas? Proteases. And gallbladder. So the gallbladder secretes lipases that break down fats. Mix it all together, and never mind, I'm not hungry either. Okay, so when you think about it, it's not very pretty. So where do starches get broken down? In the mouth. So amylase is an enzyme in your saliva that breaks down polysaccharides, so the starch. Okay, so glucose homopolymers. So these are the glucose homopolymers of plants. So starch is actually a combination of two different homopolymers of glucose. The first is amylose on the top, which is a linear homopolymer of glucose. And all the connections are alpha 1 to 4 O-glycosidic bonds. So remember when we talked about disaccharides? The O-glycosidic bond is what's connecting the two monomer sugars. And so it's an alpha. It gets trapped in that alpha position. If it's between those, the alpha, so the 1 carbon and the 4 carbon. And it's just the same all the way along. So every connection is alpha 1 to 4. So remember we talked about reducing end and non-reducing end. So what does it mean that this is the reducing end? So it can open up. And so unfortunately what they should have done, so instead of writing reducing end there, they should have maybe put an OH to show that it can open up, be in equilibrium with its linear form. But the rest of it, so at the end, it's going to end with OH there. I think they're trying to show that it's not an infinite polymer, but it's longer than just those 4 monomer units. Okay, so the reducing end has a free hydroxyl group at the anomeric carbon, so it can open and close and become oxidized, and the non-reducing end doesn't have that. Okay, and so amylose is a linear homopolymer of glucose, and amylopectin is the branched form of glucose homopolymer. And so what do we mean branched? So in addition to the alpha 1,4 glycosidic linkage that you see in amylose, there's also this branching. So now this alpha 1 anomeric carbon is attached by an O-glycosidic linkage to the hydroxyl on the C6. So remember we said there's greater diversity of connection in saccharides, polysaccharides, compared to peptides. So if you have 2 amino acids, there's only one way they can go together. But in sugars, basically you can make a connection to any of those hydroxyl groups. Okay, so the alpha 1,4 makes it a linear strand, but if you have a mixture of alpha 1,4 and alpha 1,6, now on this glucose monomer we have 2 glycosidic bonds, so that's where it branches. Does that make sense? So now starch is a combination of the linear amylose and the branched amylopectin. So how do you think that might be advantageous for this as a stored fuel? If you were going to design an enzyme that was going to come in and take off one glucose monomer at a time, would you start from the non-reducing end or the reducing end? Sorry, I did that backwards. The reducing end or the non-reducing end? Non-reducing end. So why, if you were designing an enzyme, would you have the enzyme work from this side? Exactly. There's a lot. So if each one of these is a glucose monomer, if you had enzymes, so we're not just probably one enzyme molecule, but probably lots of copies of that enzyme, they could all act on that starch at the same time. Okay, so you have all these enzymes coming in and going, doop, clipping off one glucose monomer from each of those strands, and so you can get a lot of glucose at once or in a short amount of time. And so again, if this starch is the stored fuel for the plant, so I can't imagine that the plant has to do something very quickly, but maybe it does. So maybe when it's growing its leaves, when it's first growing, it has to, grow a lot at once. And so it's advantageous to be able to get a lot of fuel in a short amount of time. Okay? And so glycogen is the animal version of that. Okay, so same rationale. It makes more sense with people, because if you say, you're going to run away from the bear and you need the glucose for your muscles, now the enzyme can come in and clip off all those glucose monomers in a very short amount of time, and you have fuel for your muscles to run away from the bear. Okay, so same thing. So glycogen looks very similar to amylopectin, except it's even more highly branched. Yes?
So does it really matter between the reducing end and non-reducing end in terms of, stability and the ease of breaking it down? The enzyme can attack one end versus the other.
So it wouldn't necessarily matter. So you could imagine that there would be an enzyme that would start at the reducing end. There just isn't. So the enzyme so the glycogen phosphorylase, which is the enzyme that breaks down glycogen to get the glucose, just happens to work on the non-reducing end. So that doesn't mean that you couldn't imagine an enzyme that would work on the reducing end. There just isn't. Yep. Okay, so where's glycogen found in animals? We already said the muscle and the liver. Okay, so what would be some advantages of having glucose in polymer form instead of in monomer form? Okay, so if you imagine the same number of monomer units, now think about the molarity of 500,000 glucose monomers versus a single polymer of 50,000 monomer units. So the molarity would be, what number did I use? 50,000 times more, right? So what does molarity have to do with cells being happy? Okay, so diffusion would actually be easier if you had monomers. What would be bad about having all the glucose from a glycogen in monomeric form in the cell? Courtney? Okay, so osmotic pressure of a cell. Okay, so if you had all the glucose of a glycogen monomer, right, a glycogen molecule as separate glucoses, the osmotic pressure of the cell would be sky high and the cell would break. Okay, because the osmotic pressure depends on the molarity, not on how many monomer units are in it. Okay, does that kind of make sense? Okay, so to maintain the osmotic pressure on a cell, it's better to have all those glucoses strung together in a polymer, where now the molarity of that goes way down. Okay, so that's why I say they're more appropriate. So that was just a hint to talk about osmotic pressure. Okay, so we'll talk about the open structure in a minute. So it's very water soluble, and part of that is water can get to it, and part of it is all the hydroxyl groups there are water soluble, right? Like dissolves like. So we have a lot of free hydroxyl groups on those sugars that we can make hydrogen bonds to water. Okay, so very soluble in water. We already talked about the multiple ends. So the enzymes can come in and, kind of a little Pac-Man thing, come in and grab all the ends off and make glucose when it's needed, but only when it's needed. And so the alpha amylases, those are the enzymes that will break those alpha 1,4 linkages and alpha 1,6 linkages. But in particular, they're alpha linkages. And we're going to see the difference between alpha and beta linkages in a minute. So we already said, where might you find alpha amylases? In your saliva. Okay, and here's just a structure of starch or glycogen. Okay, and what do you see about that structure on the right? It's helical. Yay, we see a helix again. Okay, so we're going to see helices in proteins, in sugars, and in DNA. Okay, and what about the handedness of that helix? Right-handed. So look on the right, try to make your hand do that. Yeah, so it's a right-handed helix. I know it looks kind of left on the left side of the picture, but if you look at the helix, it actually makes a right-handed helix. So it's a right-handed helix. So again, another similarity, another way to file it in your brain. And so the handedness, or the turn to it, is a consequence of how those alpha 1,4 linkages just have a pre-rotation to them. Okay? And so here's my little baby demo. Don't get too excited. So that's just water, and that's starch, more or less dissolved in water. I should have heated it up a little bit. And so do you guys ever do a starch test in biology lab or anywhere? Okay. Again, don't get too excited. It's a kind of a lame demo. So this is iodine. I think I just got it from the grocery store, the same kind of thing you'd put on a cut. And so it has kind of a golden color by itself. But if you put it into a solution of starch, it has a nice kind of blue-black color. Okay, I don't know if you can see that from back there, but it's definitely not the same color. It's kind of a bluish, it's a bluish-black. Okay, and that is a consequence of the helical structure. So the iodines line up, and their electronic structure changes enough when they're lined up that way that the color changes. Kind of cool. And so my funny iodine starch story. So you guys were, so how old were you on September 11th, 2001? 11. Okay, young, 10 or 11. Okay, and so when that happened, of course, everybody was on heightened security, all kinds of crazy fears and panics going on. So my birthday was in October, is in October every year, and my sister was living in D.C. at the time, so everybody's freaked out in D.C. There's letters going back and forth with anthrax and the letters, craziness. She sent me a skirt for my birthday from the post office where anthrax letters were received. So I always called it my anthrax skirt. And then I wore it to work and got bleach on it, and I was very sad that I had to throw it away. But the starch story, not the anthrax, well, it is related. So soon after that, the chemistry department gets a call from campus police saying, there's a white powder on the road outside. Do you think it's dangerous? And I'm like, yeah, I don't think anyone's going to terror attack Wake Forest or Winston-Salem. But sure. So they were like, so what chemical test can you do to figure out what it is? And we're like, I don't know. So we're all, a bunch of us are standing around going, how many PhDs does it take to figure out if it's anthrax or not? Even though we were sure it was not anthrax. And I'm like, it's probably flour. There was white stuff on the road. I was not a runner at the time, but in retrospect, so the punchline is people put flour on the road to mark a race course, and we just happened to see a big pile of it. And so we're like, well, I think it's probably flour. So we had some in a little petri dish, which of course, if it was anthrax, we would have been dead. And so we're like, I said, well, add a little bit of water, and if it looks like it's making gravy, then it's probably flour. And then we're like, there's some test that you can do of flour. What's that test? And then all of it all at once, a bunch of us were like, iodine, starch test. So sure enough, we made a little paste, made a little solution, added iodine, it turned blue. We're like, yeah, it's flour. So we saved the day and reassured everybody it wasn't anthrax, but really, it wasn't that scary. So, story I have about starch. Okay, so starch makes this right-handed helix. with this open structure where there's a lot of free, I don't know if you saw that, so a lot of free hydroxyl groups ready to hydrogen bond to water, right? So we can hydrogen bond to water, and so that makes it very soluble, okay? In contrast, so we also have beta-linked glucose polymers. So same thing, homopolymers of glucose, but now instead of being soluble in water, they're insoluble, and those are chitin and cellulose. Okay, so chitin is the hard outer shell on lobsters and beetles and things, so the exoskeleton, and cellulose. So chitin is the structural version for animals, and cellulose is the structural version of glucose homopolymers for plants. So the undigestible part in plants that gives things fiber. Okay, and so those are beta-linked homopolymers. So here is the beta 1,4 linkage, so same glucose monomer, but now when the enzyme that's going to make that homopolymer links them together, that anomeric carbon has its hydroxyl group in the beta position instead of the alpha position. Okay, so that enzyme selects for those beta hydroxyl, C1 beta, and links those together. And so now what's the difference? Well, can you see maybe in cellulose that they're kind of flat sheets? Okay, so instead of having that twist, we have a very flat connection just based on the geometry of the alpha linkage versus the beta linkage. And maybe you can see that there's more hydrogen bonds internally. So all those are hydrogen bonds. And so what do you think that means about the solubility of cellulose compared to starch? Solubility? Lower. Okay, so cellulose is not particularly soluble. It's the stuff that's left over. It's how we make paper, right? So if you get plant material and kind of smash it up, the cellulose is what doesn't dissolve, and so what we make paper out of. And the reason it doesn't dissolve, even though there's lots of hydroxyl groups there, is that they're already busy making hydrogen bonds, and so they're not really available to dissolve in water and make hydrogen bonds to water. Okay, so you have a relatively insoluble molecule because of those linkages, and so that's why cellulose and chitin is structural instead of fuel. So could there be enzymes that break those beta linkages? Sure. So where are those enzymes found? The bacteria in the guts of cows and termites. Okay, so termites eat wood, but it's really the bacteria in them that's breaking it down. Same thing in the guts of ruminant animals like cows. So we can't, so even though, we can cut a lot of grass, cut a lot of plants, or look at a piece of paper and say there's sugar there, we should be able to use it as fuel. We don't have the enzymes to break it down and get the glucose to use as fuel. Okay, but cows and termites do. So what are the advantages? I don't know if these are really advantages, but what are some features? So we already said that. So we can't eat paper or wood, even though there's lots of theoretical fuel there. And so what could we do with all this cellulose? Okay, so another kind of environmental question is, instead of relying on the Middle East or somewhere else for fossil fuels to fuel our cars and our power plants and everything, could we use ethanol in our cars and then decrease our dependence on foreign oil? So in theory we could, but where are we going to get that ethanol from? So if the ethanol comes from corn that we could feed the world with, not so great, right? But could we make ethanol from something like switchgrass or some cellulose, something that isn't a natural food, but if we had the right enzymes, so could we get those bacterial enzymes like the ones in termites, break down the undigestible cellulose, use that as a source for ethanol, and then maybe we're kind of killing two birds with one stone. We're not stealing the corn from the food supply to make ethanol to ship all over the country for fuel. Okay, so that's just an interesting environmental question. Okay, so don't panic, but that's basically a Ramachandran plot again. Okay, so you don't need to know the names of these bonds, although they are phi and psi bonds. So again, this is just to make the comparison to something you know, which is the phi psi bonds around the C alpha. Okay, so same idea. Just substitute C alpha for O glycosidic bond, and we can describe the rotation of those bonds. Okay, so now the rotational angle will describe what the energetic favorability of that orientation is. And so this is kind of like a three-dimensional phi-psi plot. Again, you don't have to memorize that, just know that we can describe the rotation of alpha glycosidic bonds and beta glycosidic bonds, kind of like we can describe beta sheets and alpha sheets. And, actually it's convenient that we name them the same thing. So the alpha linkages make a twist like an alpha helix. The beta linkages make a flat structure like a beta sheet. Fortunately, they match up, or else it would be confusing. Okay, and again, that's not to memorize, that's just to give you an example of, and remind you that some of these things you know already. So we just start glycogen and cellulose and chitin. And so those talk about the alpha linkages and the beta linkages, kind of what size range they are. You've probably also heard of agarose. How many of you have used agarose in lab? Okay, so agarose comes from algae. So it's the cell wall material in algae. So if you've probably never had to buy agarose. Does anyone do the purchasing in their lab? Agarose is crazy expensive. Oh, you do? Agarose is crazy expensive because it's not made synthetically. It's purified from algae. Okay, so it's like $100 for a little bottle of agarose, even though we use it like all the time, and it would be nice if it was cheaper. Okay, so agarose is a polysaccharide that we get from algae. So if you haven't ever used it before, it's convenient. So it's kind of like making Jell-O cubes. Okay, so you heat it up and it dissolves, and then when it cools, it solidifies. The really cool thing about agarose that's not like Jell-O is you can heat it up again, and it's liquid again, and then cool it, and it's solid again. So it's its thermal properties are related to its polymeric properties. Okay, and so it's used to separate DNA fragments based on size. Yes, Katie.
What's the, because like in our lab we use agar, and when I use it for it's powder, so is that just...
So agar is another sugar. I don't know why they're named so similarly.
Oh, so they're, like, it's not really expensive.
Oh, and agar's actually expensive too, but not as much as agarose. Yeah, so agarose is a sugar. I should know what agar is.
I mean, we mix it with like yeast and starch.
Right, so it's to make the liquid media of a flask into solid for a petri dish plate, right? So it's just to...
For fly food.
It's for fly food? Oh, cool. We use it to make a solid media petri dish plate on which we can streak bacteria.
It's unpurified.
It's unpurified agarose? Really? Okay. Awesome. Thanks. I'm sure I've been asked that before and never bothered to look it up. So, thank you. Okay, and then we're actually going to talk about peptidoglycan in a minute, but you don't need to know the other ones. Okay. Anyone want a 1 minute break? Stretch your legs. Okay. Take a minute break.
I think I missed that.
Okay. So were you here when we talked about alpha helices?
I was, but I was
Not fully there. Okay. So the handedness. So that one actually looks left-handed, which it shouldn't, so we won't look at that. So if you hold your right hand, can you see how it would wrap around, kind of going from bottom to top, in the same way that this wraps around? So it kind of wraps around like that. So kind of like a hand.
Okay, so it's... For me, that looks like it's doing that, but I could be...
Unless it's upset. Yeah, it's supposed to be right-handed, so let me think about why it doesn't look that way. Unless
It's doing that, right?
it's doing... Yeah, it is actually. You're right, that drawing is a left-handed drawing,
Okay.
but whether they're drawing it in that direction... Let me check.
From
Yeah.
here, it looks right-handed. but from
Oh,
because it's upside down.
yeah.
I don't know.
Yeah, because you're looking at it that way. Yeah, let me look in the textbook and see if they describe that they drew it upside down. It is a right-handed helix. So we'll try to figure out why the picture doesn't look that way. Okay. So we spent a lot of time talking about proteins. So you know everything there is to know about proteins, more or less. And we just talked about sugars. Now we're going to talk about the combinations of proteins and sugars that exist in cells. And so we're going to talk about 3 different categories. And fortunately, they're named in a way that hopefully is descriptive of how they're made up. Okay, so we're going to talk about each of these in order. So I'm not going to slide, but peptidoglycans. So you can see it has the word peptide in it. Whichever in each of these names comes second, that's what there's more of. Okay, so peptidoglycans are predominantly sugars with small peptides attached to them. Okay, proteoglycans are predominantly sugars, but when they have something attached, it's not a small peptide, it's a big protein. But still, the majority is sugar. Glycoproteins, predominantly protein with small amounts of sugar attached to the protein. Okay, peptidoglycans, small peptides attached to sugar. Proteoglycans, protein attached to sugar. Glycoprotein, sugar attached to majority protein. Okay, and that's just kind of a summary. So the first is peptidoglycans, and so you've probably heard of these, even if you don't know what their structures are. Okay, so the peptidoglycan is what makes up a bacterial cell wall. Okay, and so all these little blue stringy things, those are the polysaccharide, so the glycan part of the peptidoglycan, and so that's all this. And so for most bacteria, it's this repeating disaccharide unit of glycnec, so N-acetylglucosamine, so you don't need to know what that looks like, and murac, N-acetylmuramic acid. Again, you don't need to know what those carbohydrates look like, but the peptidoglycan bacterial cell wall is just this repeating one after the other alternating heteropolymer, but in a defined order. And then the peptido part comes in because that's what links one heteropolysaccharide to the other. So if you just had the strings of sugar, there would be some stability, but not as rigid. So when we cross-link one sugar long string to the other sugar long string, we do it with a little peptide. Okay, so that's why it's a peptidoglycan. Most of the molecule is sugar with just little peptide connections. Okay, and so in these peptide parts of the peptidoglycan, we're going to break all the rules of everything you know about proteins. Very inconvenient. Okay, so what we've learned about proteins is they use L-amino acids exclusively, right? They're made on the ribosome, and they're only linear peptides. Okay, so remember we said one difference between polysaccharides and polypeptides is there's only one way to connect peptides to each other, and we don't get branching. So I don't know if I said this explicitly, but if we have a polypeptide, so if you're only connecting the carboxylate group to the amino group, you can never get a branch point. There's only one way to connect it. So we only ever have linear proteins. True? Even if we didn't say it explicitly, does that make sense? So the disulfide is not a branch. Good. Good catch. So a branch would be so a peptide bond that just is not in. So we're going to see an exception to that. So I think I would say that a disulfide is not a branch point because it's reversible. So you have it there, so you can switch it back and forth pretty readily. It's not inherent to the connection between the amino acids. Okay, I bailed myself out of that one. So other than the example of disulfides, proteins are not branched. So here, maybe you can already see that's not a linear peptide. Okay, so if all these are amino acids, there's a branch point there. Okay, so in the peptide pieces here, we're going to see that there's D amino acids, there's branching, And so what that maybe would make you think is that it's not made on the ribosome. And in fact, these peptides are not made on the ribosome. So they're not genetically encoded in the sense of there's a DNA that makes a message that is translated on the ribosome to make a peptide. So all kinds of craziness. So if we zoom into the chemical view, so again, the black is the glycan pieces, and the blue and the red are amino acids, peptides. Yes, Jessica.
So you said there's D. Oh, never mind. It says L too.
Yep. No, so it's both D amino acids and L amino acids. So here we see D, and there's all kinds of craziness. D alanine. So there's L lysine. Those are all L glycines, L alanine, and this is D isoglutamate. What's D isoglutamate? Okay, so it's an amino acid. So it's D, and we're going to see that we have weird connections that give us branching. Okay, so here's a branch point. So is that bond an amide bond? Sure. So an amide bond is just this. Okay. Is it a peptide bond? Not exactly. So that bond is called an isopeptide bond. Okay. And in fact, this bond here is also an isopeptide bond. So what is an isopeptide bond? Say it again. No, that's kind of a red herring. So it wouldn't have to be between a D and an L. These happen to be, but they don't have to be. Okay, so it's an amide bond between amino acids, but not exclusively using the alpha amino group and the alpha carboxylate group. Okay, so where is that amine coming from in that isopeptide bond? Is it coming from the alpha amino group? No, it's coming from the side chain. Okay, so another way of defining an isopeptide bond is it's a bond between amino acids, and one of the amino acids in that isopeptide bond is using its side chain group. Okay, so lysine has a side chain amine, so that could be in an isopeptide bond. Are there any other amino acids that have an amino group on the side chain? So arginine does not have a free amino group, so don't get tricked. So yes, there's nitrogens there, but it's not a primary amine, so it can't be in an isopeptide bond. So lysine could be. And what amino acids have carboxylate side chains that could donate a carboxylate to an isopeptide bond? Aspartic acid and glutamic acid. Okay, so for an isopeptide bond, one of the partners in that isopeptide bond must be lysine, aspartic acid, or glutamic acid. Okay, so it could be the carboxylate group of glutamic acid to the alpha amine of any other amino acid.